Hi everyone, it's Becca Pretzel, and I will be sharing my presentation for Bio305 Lab on coronaviruses and the SARS-CoV-2 pandemic of COVID-19. So I'll just go into a couple general characteristics. I'll start with a reminder from Johns Hopkins University that COVID-19 is the disease and SARS-CoV-2 is the virus itself. The current coronavirus belongs to the beta coronavirus genus. Out of the alpha and beta, we're talking about the beta. It is fairly spherical in shape, as I'm sure you've seen in a lot of the pictures that have been recently released. The average virion diameter, as would be expected for a microparticle like this, is 80 to 120 nanometers. And it has a helically symmetric nucleocapsid. So we'll get into a little bit more specifics of the molecular biology of the coronaviruses in general, and we'll talk about some of the main proteins. So one of the first main proteins is the spike protein. It is a glycoprotein, and it is primarily involved in controlling receptor attachment and fusion with the viral cell membrane, the viral host cell membrane. So, when it's trying to infect, and in terms of transmission, this is a big player in that role. We'll talk more about that later, but just a little overview of the proteins themselves. The next is the membrane protein, or M protein. So here's a little bit more blown up cartoon image of that one. It also is a glycoprotein. One of its primary roles is providing shape of the virion envelope, which it does along with the help of the next protein. We have the envelope protein, kind of obvious given its name. It's an integral membrane protein. Here's another blown up image of that little cartoon image. It is believed to be involved in a lot of other things, but obviously its main function is to hold that structure of the envelope. And then the final main protein we'll talk about is the nucleocapsid protein. Again, here's another blown up image of that. It is a phosphoprotein. So the phosphorylation of this studies have shown that it may lead to association of N with intracellular membranes. But we know also that it forms helical nucleocapsid structure, so very involved in keeping that proper structure. And then it binds RNA, possibly for transcription regulation, and we'll talk more about that later as well. So just a small overview of those proteins. Some more molecular characteristics in terms of the genome. It is positive sense, this is also known as the sense strand. So this is what will encode RNA and the genetic information for making proteins. The opposite, what could the other viruses be? That then would be the negative or antisense strand, just so you have a general awareness of that. This genome is non-segmented in single-stranded RNA. And in the picture on the bottom, you can see some of the cis-acting arrangement of the RNA. You can't really see the arrangement in terms of a picture, but you can see the hairpin and the pseudoknot structures being pointed out at the bottom there. I'll show an image later where you can see it a little bit better. The genome also contains multiple open reading frames, a lot to be translated, and then the genome is involved in assembly. One of the papers pointed this out because the progeny genomes are incorporated into progeny virions. So with the generations being passed on and spread. We'll go into the replication now a little bit more in depth. Much about it is still unknown. This is an area of potential study to learn more about coronaviruses, or even specific coronaviruses such as SARS-CoV-2, especially right now. It has genomic and subgenomic RNA. So the subgenomic RNA is then what serves as mRNA for the structural and accessory genes. It has a leader and body TRSs, which are transcriptional regulatory sequences. So the leader and one of the body TRSs fuse during production of the subgenomic DNA. And then RDRP, the RNA-dependent RNA polymerase, temporarily stops at one body transcriptional regulatory sequence. So it stops at one of those, they call it TRSB. And then it can do two things from there. So either it can continue elongation of a new body TRS, or it can amplify the leader TRS, which is at the 5' end of the genome. So because of this strand jumping, it has a couple options where this RNA polymerase can go. both homologous and non-homologous recombination are possible. And this can lead then to greater viral diversity and then obviously possible viral evolution from this fact. So here's that picture I mentioned earlier. You can see the stem loop structure and the pseudoknot. You can see it looks like even a hairpin. Oh yeah, hairpins are on the right. The stem loop and the pseudoknot at the 3' prime end overlap, which that you can see where they're located in the first picture, the image on the left. And this has been proposed to be involved in regulation of different stages of RNA synthesis. And just one more thing I'll point out, the red in this image highlights the structural genomic uniqueness of the current coronavirus. More specifics of, the uniqueness of the current one will be talked later, but I just wanted to point that out so you can see that in terms of the genome right now. So then we move on to the translation of those main structural proteins. So S, E, and M proteins will be translated. They are then incorporated into the endoplasmic reticulum and then into this endoplasmic reticulum Golgi intermediate compartment. At that point, the genome becomes enclosed in the N protein capsid. Then the N protein capsid enters that ERGIC, the endoplasmic reticulum Golgi intermediate compartment. And there at that point, we have the mature virion. So then it can continue in the secretory pathway and be released via exocytosis. So then we can move on to the method of infection. Once we have those particles that are ready to infect other cells, the S1 region of the S protein will interact with the host receptor. So this image is a blown up cartoon image of the S protein. So that top portion is what interacts with the host receptor. Here's a table of a lot of different coronavirus receptors. And one of the main receptors here we have highlighted for HCoV or human coronavirus, as you can see, is angiotensin converting enzyme 2. And that was one that we discussed in one of the papers we recently read and discussed with each other for this class. So I just wanted to point that out because we're all familiar with that. So going back after the attachment to the receptor, a protease, often TMPRSS2, prompts acid-dependent cleavage of the S2 portion of the S protein. So that bottom part then will get cleaved and they will be two separate pieces. But that cleavage exposes a fusion peptide, which allows formation of a 6-helix bundle. And then that bundle promotes fusion of viral and cellular membranes. Once they fuse, the viral RNA can then be released into the host cytoplasm and we have infection. And the image here is showing kind of the fusion with the receptors and how it's entering the host. This is one of the first pictures. It's a TEM image from Hong Kong, one of the first images that they received of this mechanism. So we'll go into a couple specifics now of SARS-CoV-2. There is more efficient transmission of SARS-CoV-2 than of SARS-CoV, the original SARS coronavirus, through active pharyngeal shedding when symptoms are still mild. So in this case, it is more contagious at that point. It has a longer spike protein. So, I can't make conclusions on whether that's better, but I feel like, this is just my opinion, might not be proper, but it makes me think that if it's longer, it's, can more easily reach receptors on host cells. They have similar or even slightly earlier seroconversion. So detection of antibodies against SARS-CoV-2 may be detected in the blood sooner than SARS-CoV. And then sites of infection, this isn't really a difference, but it's more like of a specific with the current coronavirus, is that the sites of infection are only pulmonary and the GI tract, while those of other SARS viruses also include the upper respiratory tract, and sometimes, although rarely, the central nervous system. The current strategies and studies, the common public precautions you've been hearing about, wearing masks to prevent aerosolized transmission, the social distancing, the hand washing, not touching your face to try to prevent spread into the mucosal membranes. You've heard all of those. They are in practice, hopefully, by most people right now. Testing includes viral through nucleic acid amplification via PCR, and then serology for the antibodies. As we've discussed in the papers, they can detect IgM, IgA, IgG, or even total antibodies. In terms of treatment, In prevention, scientists are working towards development of vaccines and drugs. And they are also testing the effectiveness of convalescent plasma, which contains the neutralizing antibodies against SARS-CoV-2 from, say, individuals who have had it and, gain the antibodies. So transferring that to other patients, they're working on that right now as well. So what the future could or should bring, this obviously includes some of my opinions, but I think a lot of it, seems to be common ideals, such as higher prevalence of common hygiene practices, like mentioned before, just to prevent pathogenesis in general and spreading diseases. Public awareness of polite practices with how to deal with pandemics mentally, physically, emotionally, and as members of a community, to be considerate about, resources and what other people are able to obtain or not able to. And then knowledge and resources for medical treatment and prevention. Definitely, we've talked about the vaccines. One other thing, I skipped over this one, who to admit to hospitals and when people can be released to prevent overwhelming facilities and resources. And one final thing I'd like to mention are potential future studies. So some things could be how to yield test results sooner to see if people actually have the disease. And then also a greater understanding of the mechanism of RNA replication, specifically on details of the RNA polymerase to see how it's replicating and see if that can in any way be inhibited. So that's all I have. Here are the works that I have either cited or consulted for my use. Thank you for listening.